Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today I'm going to be going over some questions regarding the cell cycle unit and some about cancer and apoptosis. With that, let's get started with question 1. So as you may know, the first sign of prophase is that the chromosomes condense. Why do these chromosomes condense? And it's so they can easily be separated without any tanglements or any hassle. Number 2, the cell elongates and cytokinesis begins with the formation of a cleavage furrow, which is accomplished by what? So it is accomplished by a ring of microfilaments encircling the cell and contracting. Number 3, the nuclear membrane is absent in which of the following phases of mitosis? And you can choose as many as you want here. So you have anaphase, telophase, or metaphase. And so recall that the nuclear membrane goes away in prophase and it comes back in telophase. And so between prophase and telophase, you have metaphase and anaphase. And so metaphase and anaphase is the correct answer. Number 4, tumor suppressor genes like BRCA1 and 2 promote anti-cancer processes like DNA repair. When these genes are disabled via mutation, the risk for developing cancer later in life increases dramatically. A female who inherits a single mutation in BRCA2 has a 50% and 20% chance of developing breast and ovarian cancer respectively. Which of the following best explains why these respective chances are not 100%? A. Individuals who are homozygous for the BRCA2 mutation do not have a functional allele. B. Individuals who are homozygous for the BRCA2 mutation can still rely on other DNA repair mechanisms. C. Individuals who are heterozygous for the BRCA2 mutation still have a functional allele. And D. Individuals who are heterozygous for the BRCA2 mutation can still rely on other DNA repair mechanisms. And so the answer here is C. Individuals who are heterozygous for the BRCA2 mutation still have a functional allele. The question said she inherited a mutation, a single mutation, and so it is heterozygous, not homozygous, and therefore she still has a functional allele. Number 5. Some studies suggest that in patients with Alzheimer's disease, there's a defect in the way the spindle apparatus attaches to the kinetochore fibers. At which stage of mitotic division would one first expect to be able to visualize this problem? Prophase, metaphase, anaphase, or telophase? And this is just a recall question. So the initial contact occurs during prophase, and this is when you would be able to first visualize the problem. Number 6. A researcher wishes to incorporate a radio-labeled deoxyadenine into the genome of one of the 2 daughter cells that would arise as a result of mitosis. What is the latest stage of cellular development during which the radio-labeled deoxyadenine could be added to achieve this result? Is it G1, G2, M, or S? Now, to ensure that the labeled deoxyadenine will be incorporated into the DNA of one of the daughter cells, we have to insert the nucleotide before DNA replication has been completed. Because replication occurs during S stage, we could introduce the deoxyadenine during G1 or S stage. Because G1 is before S stage, the latest point the deoxyadenine could be added is the S stage. Number 7. Cancer cells are cells in which mitosis occurs continuously, without regard to quality or quantity of the cells produced. For this reason, most chemotherapies attack rapidly dividing cells. At which points in the cell cycle could chemotherapy effectively prevent cancer cell division? You can choose as many as you want out of S stage, prophase, or metaphase. And it would be all 3 because it's rapidly dividing and it's going through all 3 stages multiple times, and so if you affect any one stage, it will affect the cancer cell division. Number 8. Which of the following is true regarding prophase? A. The chromosomes separate and move to opposite poles of the cell. B. The spindle apparatus disappears. C. The chromosomes uncoil. Or D. The nucleoli disappear. And so the chromosomes separate and move to opposite poles of the cell in anaphase, and the spindle apparatus disappears and the chromosomes uncoil in telophase. This leaves the correct answer, which is the nucleoli disappear, and this happens in prophase. Number 9. Which of the following cell cycle checkpoints is primarily responsible for ensuring the DNA is not damaged before replication begins? And the answer here is G1S checkpoint. Recall that replication begins in the S phase, and so the checkpoint before that is primarily responsible for ensuring the DNA is not damaged, and it does so with the help of proteins like P53. Number 10. A researcher mutates a gene that causes the P53 protein to be non-functional. Which of the following outcomes is most likely? A. Enhanced apoptosis in damaged cells. B. Arrest of the cell cycle at the G2 phase. C. Uncontrolled cell division. Or D. Increased stability of microtubules. And so P53, as you know, is a tumor suppressor that is key in halting the cell cycle in response to DNA damage. And so if you have no P53, you won't halt the cell cycle and you won't cause apoptosis. So A and B would not be right, and there's nothing about stability of microtubules that's related to P53. And so the correct answer would be uncontrolled cell division, C. Number 11. Which of the following best describes a proto-oncogene? A. A gene that inhibits cell division when activated. B. A gene that when mutated promotes cell division. C. A gene that induces apoptosis when overexpressed. Or D. A gene that repairs DNA mismatches. And so recall that proto-oncogenes are just normal genes that are involved in cell growth and division, but they are frequently mutated or overexpressed. And when they are mutated or overexpressed, they become oncogenes. And so the answer here is B, a gene that when mutated promotes cell division. Number 12. Which of the following scenarios would most likely trigger apoptosis? Completion of mitosis, telomerase activation, DNA damage beyond repair, or presence of growth factors? And this is an easy one. It is DNA damage beyond repair. Number 13. During which of the following portions of the cell cycle is DNA polymerase most active? M, S, G1, or G0? And so the answer here is the S phase, as this is when the cell replicates its DNA, and this is when you would need DNA polymerase. Number 13. During which of the following portions of the cell cycle are mRNA and proteins mainly produced? M, S, G2, or G1? And so by asking about mRNA and proteins, it is asking about gene transcription and protein translation, and this occurs mostly during growth and organelle replication in the G1 portion. So G1 is the correct answer. Number 14. Chromosomes are often made visible using gymesis staining. This stain shows specific banding patterns for chromosomes and helps scientists organize them under a microscope. Considering that chromosomes are the standard unit of organization for a cell's DNA, during which phase of the cell cycle would chromosomes most likely be visible? Would it be in the M phase, S phase, G1 phase, G2 phase, or are they visible during all portions of the cell cycle? And so chromosomes are most likely to be visible when a cell is organizing its genetic material, as it would be just before it undergoes cytokinesis and becomes two daughter cells. So the answer is M phase. Now S phase is a tempting answer, as this is when DNA is duplicated, but S phase only encompasses the enzymatic replication of DNA and not its organization. Okay, number 15. There are several methods for analyzing the number of cells undergoing proliferation. Cells or whole organisms can be treated with BirdU, which is a uracil analog. This is incorporated during DNA synthesis. Cells or tissues can be fixed, and BirdU can be detected using BirdU specific antibodies. Similarly, cells can be fixed without any pretreatment, and proliferation can be detected by antibodies specific for MK167 or PH3, which is phosphorylated histone 3. MK167 can be detected at all active phases of the cell cycle. Remember that this does not include interphase. While PH3 can be detected during mitosis only. And so which detection method should be used to detect the greatest number of healthy cells? BirdU, PH3, MK167, or will each method detect the same number of proliferating cells? And so the answer is BirdU, And so because cells can be treated with Bourdieu in advance of analysis, a large window of time can be analyzed. MK167 and PH3 only measure proliferation at a snapshot of time. Bourdieu analysis will essentially provide a total number of cells that are capable of proliferating since the initial treatment with Bourdieu. Any cells that have undergone DNA replication since its addition will be detected, while quiescent or dead cells will not be detected. In contrast, MK167 and PH3 will show only cells that are in the act of proliferation when the sample is taken. Normally replicating cells that are simply in interphase will not be detected, even though they are healthy. Okay, and last question. Which of the following factors might cause cell cycle growth arrest? DNA damage, failure of the chromosomes to line up on the equatorial plate during mitosis, lack of appropriate growth factors, or all of the above? And the answer is all of the above. And with that, that's it for today. Next time, we'll start with the digestive system.